professor of pharmacy here at the University of North Carolina in Chapel Hill, and he's agreed to talk with us about some of the work he does and the roles that information plays in his research. So why don't we start with just a little description of what's the kind of research you're interested in? What do you do?
Thank you, Gary. So I would qualify myself as an information scientist, or I've become an information scientist with background in chemistry. And my interests are in the area of computer-aided drug discovery. And the type of information that I analyze relates to chemical structure and their functions in a broad sense. So my interests are in developing databases, creating databases, analyzing databases that contain empirical information about chemical entities and their biological and physical properties, and then analyzing those databases to establish trends in data, extract those trends, and then convert trends into predictive models that would allow us to make predictions of novel chemical entities that have desired properties, desired functions. So that's, I guess, my angle of using information science as applied to chemical databases.
This is fascinating to think about the notion of structure and then function. How do those come together? How do you make those connections?
So structure and function are inseparable. And in a physical sense, chemical structure is some sort of absolute description of a chemical matter. It exists. And as we exist and breathe, chemical structures exist and have certain functions. Sometimes the functions become obvious only if you touch them. If a drug molecule is stored in a pharmacy store, it doesn't do anything. When you take a drug, it makes you feel better, hopefully. But each structure potentially has certain function that's affiliated with it. In most cases, the function is known. In many cases, it's unknown. The databases which store information about structures and their properties is the database of databases of knowledge. What we do is extract additional knowledge from these databases, again, extract sort of trends that relate in quantitative sense, structure and function, or structure and property. The area of computational drug discovery, that's an area of my interest, is concerned with facilitating the empirical process of discovering new drugs, minimizing the amount of experimental effort that goes into chemical synthesis, that goes into experiments that evaluate properties, biological functions or biological activities of chemical structures, making the whole process of discovering new medicines faster.
So these empirical investigations depend on certain kinds of parameters. So if excuse me for not fully appreciating all of the structural properties, but one of the things I think we've talked about in the past has been angles, either within. Are you talking about angles within folds in a protein structure, or actual chemical bonding angles in at the molecular level, or both?
Sort of both or neither, if you will. I've been talking in a rather global sense about molecular structures and their functions. Now, there is a need that is in part at least met in organizing data. Data in the chemistry world are organized around how we describe structure and how we create ontologies of structures, if you will. And so people typically separate low molecular weight organic molecules. That is a domain of synthetic organic chemistry and synthetic medicinal chemistry, and macromolecular structures such as proteins and nucleic acids, which frequently serve as targets, molecular targets of low molecular weight drug action. In the functional sense, the separation sometimes is inaccurate because, as I mentioned to you recently, many novel drugs are so-called biologics, which are proteins that are used as drugs. So drugs are not limited to organic molecules. per se. So what I was talking about is in a very general sense relating molecular structure, regardless of its size and specifics of its organization, to its function in a global sense. In a more distinct sense, we are interested in my lab in analyzing the relationship between low molecular weight chemical structures, where parameters that we take into consideration are bonds and bond angles, sort of fine resolution description of molecular structure, and its specific biological activity as anti-cancer agents, for instance, that reduce the size of tumor in experimental tissues, as well as in a separate set of investigations looking at some macromolecules where this more minuscule description is important, but in computational sense becomes frequently and rapidly impractical. And there we're looking at more global parameters, bigger parameters of molecular structure, and analyzing them at the level of individual amino acids contributed to the overall protein structure. And then relating sort of global macromolecular structure of large molecules to their biological function as enzymes or biological receptors. So there are these sort of differences between the type of activity or function that we analyze with respect to small molecules and that with respect to macromolecules. Right? So again, sort of reiterate once again, with small molecules we may be looking at a set of compounds with relatively simple structure that have been tested in experiments that would indicate that they reduce tumor or eliminate tumor. And so that would be a structure anti-carcinogenic activity, anti-tumor activity of compounds. And we would try to extract trends and design new compounds that would be stronger anti-tumor agents. In case of macromolecules and protein structures, we might be looking at a collection of enzymes that collectively promote tumor growth. And in that case, we would identify determinants that make them active in promoting tumor growth, and then would rely on them as specific targets for which we need to develop new low molecular weight molecules.
So those determinants in those large molecule senses, you can sort of knock them out, if you will, and then see what the effects are. Is that an approach?
In that sense, you could use tools of molecular biology and knock out the entire enzyme, the entire molecule, the entire gene, essentially eliminate the entire gene and establish its relative significance in tumorigenic process. Or you could use more subtle tools and mutate an amino acid residue, essentially a single residue or a few residues in any given protein, to establish whether or not this residue is significant in functioning this enzyme, of this enzyme. And then use this information in targeting drugs more specifically to certain areas of a macromolecule.
So the combinatorial explosion has just got to be great in terms of all the various things you could try.
And what I know that you're interested in information and informatics aspects of science, and this discipline that I'm in, that is studying relationship between molecular structure and its property in a global sense, has become an extraordinary reach as far as the amount of information that has been developing. There are multiple publications that basically tell us how much the space of data has changed in the past decade, essentially. But so-called high throughput technologies have been generated just enormous amount of data about both structures, just the sheer number of structures that are synthesized or characterized these days, from genomes to organic chemical molecules, has been growing more than exponentially, incredibly, in the past couple of decades. And for many of sort of pure structure generating tools and technologies, there are also tools and technologies that study empirically what their function is. So it's really a great area to be in for an information scientist in terms of organizing this information, creating these huge matrices of information, and then especially analyzing these matrices and deriving trends
Right.
So it's an enormous amount of novel science that can be done by specialists in information retrieval, computer science, databasing, data mining, statistics, biostatistics, software development, graphics, visualization of complex data. It's totally new world in this regard.
It's fascinating. I know that in addition to the theoretical work that you do, you and your lab have produced several software tools that other people use to do their discovery. Could you describe a little bit of some of these tools, like SNAPP, I think is one of them, or Motif Finder, the various tools that you've developed?
So I guess it sort of relates to how we view what we develop. As scientists, I think it's important to not only discover but deliver discovery, if you will, and so tool sharing, method sharing has been important for us. When we ask questions and then try to find answers, our answers translate into software that we initially use inside the lab and then make it available on the web. So SNAPP server that you've mentioned is using fairly sophisticated methodologies borrowed from data mining, and specifically graph mining and computer science, and applying this methodologies to protein structure, representation, and analysis. And what this tool does is it analyzes protein structures to identify their cores of stability, identifying them, visualizing them, pointing people toward where or which specific amino acid residues are responsible for protein stability. Now, if protein is unstable, certainly it cannot function. So that immediately translates into some sort of predictive tool as well. Yeah. And so that's a web-based server that we have been developing and polishing in the past decade. In the area of computational drug discovery, we've been developing many algorithms that explore massive data that relate chemical structure and its activity. Chemical structure in our hands is represented by what's called molecular descriptors, which are multiple attributes or descriptors of chemical structure. And then, of course, we know from the experiment what kind of activity these chemical structures have. So we're using a lot of correlation type modeling that relate values of chemical descriptors to values of biological activity. And again, these tools that we've been developing, we are making them available in the form of software that runs on web server so that people could upload their data sets, analyze their data sets using our techniques, and then use the results as, again, hypothesis generators.
So these descriptors, these molecular descriptors, do they come, or the experiences that you mentioned, do they come from the literature, or is that based on some theoretical principles?
In part from the literature, in part developed in our group.
Okay.
In principle, if you want to analyze a problem, you need to formalize it. That's pretty generic. In the context of our research, when we formalize a problem, we have to formalize the description of molecules. Now, chemical structures can be characterized in a number of ways. Experimentally, they're characterized by various physical parameters: melting point, freezing point, structure obtained from structural analytical methods. In cheminformatics, which is one of my major disciplines, we use theories and methodologies developed in mathematical chemistry, chemical graph theory to derive parameters of chemical structures that quantitate the structure. So they're essentially multiple equivalents of a structure, and together they characterize various facets of the structure. So that's the part of pre-model development. This is the part where we formally describe molecules. Once this part is concluded, which is a discipline on its own, then we use the results of this to when we have mathematical parameters of the structure. We could use mathematical and statistical techniques to establish correlations, essentially correlations between numbers. I guess, for a layperson, the closest analog, and I use this analogy when I begin my classes, is that the data matrices that we analyze are not that much different from those that Wall Street analysts use when they are trying to predict the behavior of stocks. For instance, when they accumulate multiple attributes of a company or various companies and then try to relate these attributes to the performance of the company. So in our case, it's attributes of chemical structure, and we're trying to forecast from these attributes what's going to happen, what's going to be the fate of a molecule as a drug.
Right. Yeah, that's fascinating. What's the role of visualization? And when you have a visualization of a three-dimensional structure, do you save it? Does it get represented? How do you sort of deal with all the data?
So it's a great question, right? So it's actually two questions, and probably even unknowingly to yourself, you've asked two different questions. So one that has been one of the major foci of a field called molecular modeling for the past, maybe now nearly 40 years, is how do you visualize chemical structures in three-dimensional space in real time? And that question was asked, and a field was developed called molecular graphics, with a great deal of contribution from UNC, Department of Computer Science,
right.
Fred Brooks and his colleagues. And that's the matter of looking at individual molecules, visualizing them, looking at how small molecules interact with macromolecular targets, and using visualization to as some sort of imaginative data support tool to come up with modifications of existing bioactive molecules to make them more biologically active. So the tools that have been developed over the years and used essentially by every pharmaceutical company is so-called molecular docking, when you essentially replicate the ship docking process except at molecular level, when you look at how a small molecule navigates itself into the active site, and then looking at what else can be done to increase the affinity of the molecule to the active site. So that's three-dimensional molecular visualization in three-dimensional space, taking chemistry into account. Now, the data that I've been talking about when we initially calculate molecular structure in the form of multiple descriptors, the analogy that I used with stock, we're basically talking about representing chemical structures in multi-dimensional descriptor space, way beyond 3 dimensions. And also not a single molecule that interacts with a few molecules that we could look at the same time, but dozens of thousands of objects that I described in hundreds dimensional space. So how do you do this, and how do you visualize this data? So that has been also an important emerging area in multidimensional data analysis, and we have been collaborating with statisticians such as Steve Marin here in statistics and operations research. People who are concerned with how you, with losing some information, but how you visualize in 3-dimensional space objects that are initially characterized in 100-dimensional spaces. So there are techniques that are used to project data from high-dimensional space onto lower-dimensional space, and then represent this data. So that has been an interesting area as well.
So this is a little bit similar to the kinds of work that people do in information retrieval, in which they take a very high-dimensional space of concepts and try to project them down into a few
manageable, interpretable.
Yes,
right. It's in this regard,
yeah.
and I think what's been happening in my discipline is characteristic of many disciplines, and what I find most exciting and interesting is transparency of techniques, making them sort of transcendent, linking very unrelated disciplines, or seemingly unrelated disciplines. There were publications in chemical literature and medicinal chemistry, computational medicinal chemistry literature, where people would borrow techniques from structural linguistics to analyze data, and I think we've sort of briefly talked about it in the past. And I find this science of analyzing high-dimensional spaces, regardless of the origin of the space, a very interesting common trend of modern science.
Yeah, people are able to do this somehow automatically, to take multi-sensory information and put it all together and determine how to act. We don't really know how to model it very well yet. Your work must generate just enormous data sets. Now, do these data sets get stored in traditional databases, or do you develop new kinds of special structures to save these things?
No, I would say that we use traditional Oracle type relational databases or database languages to store information. I think there are, again, many current disciplines where web analysis is one of them, where people deal with huge massives of data, or clinical data. So I think the size of databases that we generate has grown and now matches perhaps weather databases, for instance. So would sort of benefit from database development or database treatment methodologies developed in other disciplines.
And is the data itself numeric, textual, visual, sensory? I mean, it's
most of the data is numeric.
Numeric, yeah.
Some of the data, especially in the area of toxicology, which is one of our areas of interest, computational chemical toxicology, frequently textual. That is, that people would describe, toxicologists would describe their observations of animal behavior in response to a toxicant. And that's something that we have difficulty dealing with because methods that we use are more formalized. Right. And that's an ongoing difficult investigation as to how you translate textual description into numerical parameters. And there are some ways of doing this, and it's probably, I mean, just based on your reaction, that's something that you probably deal with a lot. Yeah. But when people say that a chemical caused animal behavior to slow down, sometimes they would use qualifier, and they would say this one had slowed it down more than other, so would then use categories then and translate wordy descriptions into categories.
Right.
And especially again in the area of what's called drug property prediction, metabolism, expression, toxicity, people frequently would categorize compounds as slow metabolizers, moderately slow, moderately fast, fast. That translates into 4 categories that leads to classification type model.
Right. Yeah, one of the big problems in the information retrieval community is if you return a set of eigenvalues or numbers to people, it doesn't mean anything. You have to somehow label the clusters, and that's where really the human side of it really has to come in.
That's actually, it triggered something that has been an interesting development in my discipline. So naturally, a lot of tools that we use are statistical in nature, and they produce models that are characterized by various statistical parameters of accuracy. Many, people in statistics have thought about dozens of various parameters of. Now, these parameters mean nothing to chemists. Chemists want to know how the new structure should look like. So model interpretation in a way that is accessible, understandable, and useful. And I think, again, this is part of information science as to how you explain what you do to not narrow specialists in your field. And, personally, I felt that we as sort of computational statistical scientists are guilty for not returning to the world what we took from the world to be successful in our disciplines. And what I mean is that people that we depend on, medicinal chemists, they produce chemical structures and they measure their property. So they give us information in a very simple form. Here's the chemical structure, and this is a value of biological activity for the structure. And faithfully, what we return are a bunch of statistical parameters of models that we create, which is completely different language from the input language that we use.
Right.
And the reason, of course, is that we've struggled for many years trying to translate information in one direction from traditional for chemists to understandable to statisticians.
Right.
Well, once you've done this, you live in your own world. And then you're not concerned, because the world is sufficiently large, so you have enough people who understand you. You're not really concerned about translating models back into the language that is understandable by people who provided you information in the first place.
Yeah, that's fascinating. I want to think about that some more, because it's like translating from one language into another and then back again, and not only
And
what
there's
gets
no
lost.
conversation. In this case, it's basically you're understanding, but you're not speaking. And
Yeah.
so I came up with somewhat self-offending term describing what we do as a narcissistic modeling.
Yeah.
And I just realized this recently.
Yeah. That's really interesting, because it relates to the kinds of interface issues that I'm interested in,
Right.
but I don't know how to do that either, and so this cries out for collaboration and
Absolutely.
Yeah. Oh, that's great. Earlier you mentioned this idea of research for evaluative research and predictive research.
Correct.
Could you say a little bit more about that? This
Right.
is.
I think it's a fine line in many respects. So, and I think it's something that had plagued our discipline for many years, and that is lack of focus. It somewhat relates to what I was just talking about as narcissistic modeling. That is why we do work in our discipline. What do we study and how we evaluate the results of our research. So when we collect information from literature sources, from scientific literature sources, we end up creating a database. Let's say that many scientists in different countries had been concerned with specific endpoint, HIV protease as an enzyme that is target of anti-HIV drug discovery. And few dozen labs have been analyzing different chemicals and then studying them and generating data on their anti-HIV activity. So an informatician would put this disparate sources together and create a large or the largest database where all compounds will be collected together.
Right.
That's a substantial undertaking and a useful one. So now we have a database. That's a creative part. So that's a synthetic part.
Right.
So you synthesize a database. Then you could query this database, and that's analysis. So if you analyze this database, you could even create a model of the data in this database, and this model might even extract some trends that relate structure to activity. But at this stage, there is no hypothesis being generated. So I regard this stage as evaluative. You evaluate data and you find trends in this data, perhaps even. But it does not yet become predictive science in a sense that you could demonstrate, that you could predict data that's not in the database yet accurately and then validate this by experiment.
Right.
So my major research interest had become in focusing on models that have demonstrated predictive power. So yes, we need to create the database. Yes, we need to analyze it. Yes, we need to find trends in this database. But up to this point, this is still sort of self-serving in a sense of analyzing the information. So I call predictive science, which I think is true for any mature scientific discipline, is when you use data to generate models of the data that actually generate testable hypotheses that in most cases, when validated by experiment, prove to be true.
Interesting. And then where does the sort of theoretical principle come in? I mean, where I empirically validate it, and then if I mathematically prove it or have a formal model, that fits in with the predictive part?
I think it's more empirical, at this stage. So data that we analyze are highly empirical in nature and highly non-mechanistically explainable, if you will. The target properties that we measure, sometimes it's a reaction of the whole animal to a chemical. And there are probably hundreds of thousands of biochemical reactions that are affected by a chemical, and we don't know many of them. We're analyzing the outcome. The sick animal had become in an animal model is not as sick anymore, or the longevity of life increased or something like this. So it's what we're building are empirical models that statistically, in statistically significant way, relate chemical structure and the outcome of its action. If we could prove empirically, and the way we do it is that we artificially create training, test, validation sets. Ideally, and that's where I've been spending a lot of time recently, is finding real-life collaborators who will actually validate my hypothesis. And so I find most rewarding and sort of find as a complete scientific exercise is when I say that my work is, or at least intermediate of my work is over. Not when I created a database, not when I created a model of this data, not even when I theoretically demonstrated that I could accurately predict something that was not used in model development. But when I actually have an experimental collaborator who tests a certain number of compounds I predicted to be active and proves that my prediction is correct.
Wow. Yeah.
That's a significant change, at least in our field, from sort of a paradigm and quality, publication quality level. And I find this is probably the most important paradigm shift. Proof. Not only generate a testable hypothesis, but prove that this is correct.
That's fascinating. It takes the traditional theoretical scientific paradigm and makes it gives it an application, and that it has to mean something.
It gives it a real proof of concept.
Yeah. That's neat. I have just one more question for you. The way you think about the concept library, it's got to be different than, let's say, my colleague in the next building thinks of library. So when you hear that term, what comes to your mind?
I think it probably has changed. If I asked the same question 20 years ago, I probably would have answered. These days, I think it's a database of information, mineable information. It's basically a learning source. That's what I think about. The word library, of course, has been used, all the time in library science, implying literature sources. People had borrowed this term to describe chemical libraries. And so for me, it's a starting point of my investigation. Library is where I go and learn and establish and develop a new library, if you will. We're talking about establishing libraries of models, right? For instance. So it's ongoing development kind of term.
Well, this has been just fascinating and very, very helpful. Thank you very much for your time, and best wishes for continued success in your research.
Thank you. It's been fun.